Once again, welcome back, student. Today we will talk about the new topic, that is genomes, proteomics, and bioinformatics. So, for your kind information, as we know that some of the syllabus are reduced in this chapter also, but we must know what are the genomics, proteomics, and bioinformatics. So, before going to start the chapter, I would like to dictate the term genomics. Genome is what? The total number of the chromosome present in individual is known as genome, and genomics is the study of presence of genome or the chromosome number of an individual in its body cell is called genomics. So genomics is defining about the number of genes, number of chromosomes, or what are the codons that are able to make the protein, and thereafter it undergoes the transcription or translation. All things are studied in the genomics. And as we know that if we are this is the double stranded structure of a DNA. So chromosome is a chemical structure, it is a thread-like structure, and chemically it made up of DNA, RNA, and protein. This is the chemical composition of the chromosome, or you can also say the chromatin fiber. Now, if we are talking over the DNA structure, double helical structure of a DNA, so all the part of DNA is not functional. Suppose I am taking this region from here to here. So this is the region which are functional. Suppose this region is non-functional. So it is known as junk DNA, and it does not code for any kind of protein formation. And this region of the DNA is the coding region, and it undergo the process of transcription or translation and then make the proteins. So this is the meaning of genomics and the genome. Now the term proteomics. The term proteomics is total number of proteins present in individual or living cells is known as proteomics. And the third one is bioinformatics. Bioinformatics, the emerging new branch of the science which has a wide scope. And here what we are doing here, these biological information are incorporated or transformed into database analysis. So these biological information is converted into database analysis with the help of the PC or the computer. So that is known as the bioinformatics. What we are taking here, we are taking the DNA. As we know that DNA is having their codon, and these codon are having the particular triplet of nucleotide. So these codon are fed into the database in a computer. So this is known as the bioinformatics. Before going to that, now we will begin the first first heading or the first content, that is gene prediction and counting. Now the term, what is the meaning of prediction? Prediction is forecasting or to analyze what is really happened or what is the cause behind this gene or the genome. So gene prediction is an approach. It is an approach to find out total number of gene count, total number of gene. Second, how these genes work, how many of codons are functional, or non-functionals. So this is the gene prediction, and these gene prediction is generally computed or it is analyzed by the two technique that we call the mapping. The word is mapping of gene and gene sequencing. There is a two technique which we are using to gene prediction: exact location of gene in the individuals, since the chromosomes are very lengthy and they are highly covalent structure, so it is tough to find out the exact number of the gene or the genome of the individual. So gene mapping and the gene sequence is a method to find out that. Before going to that, you must know that HGP, that is the Human Genome Project, that so Human Genome Project was launched in the year 1990 and it is over by 2003. So this is 13 years of the project, and the main objective of this HGP was to find out the total number of the genome or total number of chromosome or the functional genes which is present in the human body, and that should be converted into the database analysis. So according to this Human Genome Project, we can exactly tell that there are about 3 billions of genes base pair. There are 3 billions of gene, or you can say the base pairs are present in the human body, but out of that the functional which is known is 20,000 to 25,000. This is the total number of gene as per the data reveals from the Human Genome Project. But exactly some of the biologists believe that this number may be 40,000 to 50,000. But now why there is differentiation from this data 20,000 to 25,000? It is due to the in silico, that is computational. Why we are computing the database analysis? So there are some error takes place or there is some changing. Due to that changing, this number reduced to 20,000 to 25,000. So this is the gene prediction. Now what is the gene counting? This gene counting is what? As again counting, that is the counting the number of genes present in individual, and it is also a not very easy task to counting the number of genes. So total number of genes or the allele present in an individual is known as gene counting, and this is one of the attributes of population genetics. And in the population genetics, you have studied about the Hardy-Weinberg principle. Hardy-Weinberg principle. According to Hardy-Weinberg principle, what happened? The frequency of allele of a particular population will remain constant, and it is based on the P plus Q square is equal to P square Q square plus 2P Q, and it always will come 1, and this is the value of the gene frequency. But in a individual, in a population, actually we are counting the one individual of a population. So there are 1,000 people, there are 1,000 individuals in a population living. So allelic frequency may be different. It is not exact that whatever the number of genes is present in one individual, it will be on the same one, because what happened, sometimes the genes are also genetic difference taking place, gene migration takes place, gene flow takes place also. So the population size or the total number of population or total number of genes present in population will be varied. And this counting is basically based on the presence of alleles, or you can say the chromosome number. Alleles are what? The alternative form of a gene. Suppose there is a gene P and its alternative form is suppose P1 or P2. So these are the allelic form. So by either by counting this allelic form or by counting the number of gene is the way to find out the gene counting. So on that ground there are tabulation or there is a tabulated data given regarding the gene prediction and the counting. For example, organisms, we are taking the organisms, then number of chromosome, then base pair number of present genome, number of prediction gene, and percentage of gene take part in protein formation or synthesis of the protein. So first one is the Escherichia coli, the bacteria, and since it is a prokaryotic, so its haploid chromosome number 1 is there, and total number of base pair is regarded 5 lakh, and the number of prediction of gene is there 5000, and 90% for the gene or genome take part in the protein functioning or synthesis of the protein. Come to the yeast, Saccharomyces cerevisiae, where the chromosome number is 16. Here is the haploid set of chromosome number. We are not telling the diploid set of chromosome number. So haploid set of chromosome number, and the base pair is 1206800, and the predicted number of genes out in this yeast or Saccharomyces cerevisiae is 6340, and percentage of the genome take part in protein is 73% only. Now let us see the Drosophila melanogaster. So that is for the fruit fly. Here the chromosome number is 4, and their base pair ranges from 175 into 10 to the power 6 to 196 to 10 to the power 6, and predicted number of genes are 13,600. You can see there very less number of genome take part in the protein. Now human, that is Homo sapien, haploid set of chromosome number is 23 only. It is expected 3 billions. Total number of base pair will be regarded or will present 3 billions, and the number of predicted genes is about 20 to 25,000. This data is as per the HGB Human Genome Project, and less than 5%. Exactly, it was told in the HGB that less than 1% of the gene take part in the synthesis of the functioning of protein. So you can see that there is a slight change in the contribution of the genome in the protein formation or protein functioning, and in human beings it is very less. You can see that on the basis of simplicity to complexity, as we going complex, the protein contribution of the genome is very less, and if organism is very small, there you can see the more contribution of the genome in the formation of the protein. So this is about the gene prediction and counting of gene. Thanks for watching.